Armed TH1 cells have a central role in macrophage activation. A number of important pathogens live within macrophages, whereas many others are ingested by macrophages from the extracellular fluid. In many cases, the macrophage is able to destroy such pathogens without the need for T-cell activation. But in several clinically important infections, CD4 T-cells are needed to provide activating signals for macrophages. The induction of antimicrobial mechanisms in macrophages is known as macrophage activation and is the principal effector action of TH1 cells. Among the extracellular pathogens that are killed when macrophages are activated is Pneumocystis carinii, which, because of a deficiency of CD4 T cells, is a common cause of death in people with AIDS. Macrophage activation can be measured by the ability of activated macrophages to damage a broad spectrum of microbes as well as certain tumor cells. This ability to act on extracellular targets extends to healthy self cells, which means that macrophages must normally be maintained in a non-activated state. Macrophages require two signals for activation. One of these is provided by interferon gamma. The other can be provided by a variety of means and is needed to sensitize the macrophage to respond to interferon gamma. Armed effector TH1 cells can deliver both signals. Interferon gamma is the most characteristic cytokine produced by armed TH1 cells on interacting with their specific target cells, whereas the CD40 ligand expressed by the TH1 cell delivers the sensitizing signal by contacting CD40 on the macrophage. CD8 T cells are also an important source of interferon gamma and can activate macrophages presenting antigens derived from cytosolic proteins. Mice lacking MHC class I molecules and which thus have no CD8 T cells show increased susceptibility to some parasitic infections. Macrophages can be made more sensitive to interferon gamma by very small amounts of bacterial lipopolysaccharide, and this latter pathway may be particularly important when CD8 T cells are the primary source of interferon gamma. It is also possible that membrane-associated TNF-alpha or TNF-beta can substitute for CD40 ligand in macrophage activation. These cell-associated molecules apparently stimulate the macrophage to secrete TNF-alpha. An antibody to TNF-alpha can inhibit macrophage activation. TH2 cells are inefficient macrophage activators because they produce interleukin-10, a cytokine that can deactivate macrophages, and they do not produce interferon gamma. They do express CD40 ligand, however, and can deliver the contact-dependent signal required to activate macrophages to respond to interferon gamma. And that's armed TH1 cells have a central role in macrophage activation. Have a great day.